Hello, I am Noah Hogan, and the topic of interest for my final project is the effects of caffeine on amyloid beta oligomerization. The reason why the oligomerization of the amyloid beta peptide is important is that this peptide is directly related to many age-related illnesses, Alzheimer's being chief among them all. Alzheimer's is an age-related disease, meaning the chances of developing symptoms attributed to the disease rise as one gets older. At age 90, there is a 30% chance of developing Alzheimer's disease throughout one's lifetime, and the chance of developing the disease gets even higher with age. Although there is not much consensus in scientific literature as to how Alzheimer's is caused, the tau tangle theory and the amyloid beta plaquing theory are the most common explanations for the occurrence of the illness. These theories dominated scientific research until very recently, when it was discovered that the oligomerization of the amyloid beta peptide could create unwanted ion channels in healthy neurons. These ion channels disrupt calcium regulation within the neural cells, causing apoptosis of such cells and further neurodegenerative diseases. Caffeine, a commonly consumed stimulant found around the world in plants and beverages, is currently being researched due to its ability to block amyloid beta plaques and delay the onset of Alzheimer's disease. Both caffeine and amyloid beta peptides are amphiphilic in nature, which means that they both contain hydrophobic and hydrophilic components. When amyloid beta peptides are released into the cell, caffeine interacts with the peptide, disrupting the amyloid fibril creation process. This reduces the amount of amyloid plaque buildup in the brain, ultimately slowing down the rate at which Alzheimer's disease develops. Previous research has been conducted to find the supposed effects of caffeine on age-related illnesses, and several different models have been used to investigate these claims. One piece of scientific literature that I found on the subject was focused on the effect of just the caffeine molecule on Alzheimer's disease in mice. This study also factored in the timescale of the administration of the drug, as they examined both the short-term and long-term effects of caffeine administration in Alzheimer's prevention. The study concluded that both short-term and long-term administration of caffeine was effective in slowing the rate at which the onset of Alzheimer's occurred, and the results even went to show that caffeine may be an effective tool in the treatment of early Alzheimer's. The diagram shows that caffeine may be effective in disrupting the structure of amyloid plaques in the brain, making caffeine a viable option for the treatment of Alzheimer's. While mice may be a good analog to observe the macroscopic effects of caffeine on the delay of Alzheimer's disease, a more thorough explanation is required to find out how the caffeine molecules interact with the amyloid beta peptides. Amyloid beta peptides have a tendency to create other amyloids, making them hard to characterize with a traditional lab setup. The beauty of computer simulation is that it provides a metaphorical computer microscope to generate insights that are not attainable through traditional means. The second study I examined used this computational microscope to examine the effects of caffeine on the aggregation propensity of the amyloid beta 16 to 22 peptide. This piece of research came to the same conclusion as the one found with the mice, but it also provided a mechanism that may be responsible for the removal of amyloid beta peptides in the presence of caffeine. The study concluded that caffeine lowers the interpeptide interaction tendencies within the neural cells, which prohibits formation of the amyloid beta plaques. While this answer was satisfactory, amyloid beta plaquing is not the only cause of Alzheimer's disease. The theory that amyloid beta oligomers form ion channels in neural cells is becoming a more prevalent pathological theory. So I want to assess the effects on caffeine on this particular phenomenon. The first step I took to solve this problem was researching the peptide responsible for the oligomerization of amyloid beta peptides in cells. The next step in the design of my project was solvation of the protein. I had to create a solvent for the protein that mimics standard biological conditions, and I had to add caffeine at different molarities to see the effects caffeine had on amyloid oligomerization. Next, I had to take the solvated molecules, perform minimization of the molecules, and create import files that NAMD would recognize and run. I would then be tasked with creating a script that would allow these NAMD simulations to run on ASU's computer cluster without errors. Once the NAMD simulations were complete, I had to observe the trajectories of the final states of the structures in VMD and perform statistical analyses to see if the addition of caffeine was relevant in minimizing these oligomerizations. Step 1 of this process involved finding the proper protein to use on the protein database and downloading the protein onto my computer. After some research, I found the proper basis for the formation of the amyloid fibrils, so I downloaded the peptide on my home computer for use in further steps. The image to the right is the image of the protein that I generated in VMD using the name of the protein residues to color the molecule. Step 2 in my project involved the solvation of the amyloid fibril and the creation of boundary conditions that would be accurate in a biological context. This was definitely the most difficult part of the research process, as it proved to be a very time-consuming and cognitively straining endeavor. First, I tried to use QuickMD to solvate the amyloid peptide in a solution of just water and salt. Between VMD installing the wrong packages, configurations options not being set correctly, and random errors that seemed to crop up out of nowhere, I had to resort to a new and more robust file builder. The second attempt I made at creating an NAMD input file involved the use of the PSF builder and Solvate plugins within VMD. These plugins did create a valid water box, as shown on the picture to the right. The problem with this is that I could not find a way to add my caffeine molecules to the water box, and there was no way to control the concentrations of such molecules. Also, I knew that my force field parameterization would not be accurate, as I do not have the knowledge in that side of computational chemistry just yet. After searching online for a better way to create NAMD input files, I found the Charm GUI website, so I decided to give that a shot. Building a solution with the Charm GUI was a fairly straightforward process. First, I uploaded the PDB file that I generated when I used VMD to create NAMD input files. After that, I was presented with several different options for editing the PDB file, but none of these parameters were pertinent to my experiment, so I stuck with my default PDB file. The next step was the step where I could parameterize my solution. I was hoping that there would be an option to add a ligand, such as caffeine, to the solution and control the concentration of such a ligand, but no options were available for such a process. Instead, other ions such as potassium chloride and sodium chloride were present, so I solvated my amyloid beta molecule with 0.15 molar sodium chloride. Finally, I selected the option to create a file meant to run in NAMD, and I further parameterized the equilibration and dynamic simulations to run with an MVT ensemble at 303.15 Kelvin. At this point, I started to realize how large the scope of a project involving ligand binding must be. Not only would I have to solvate the amyloid beta peptide in a solution mimicking the conditions found in the human body, I would then have to add the caffeine molecules and parameterize such molecules to behave as they would in the human body. I would have to create NAMD configuration files beyond the scope of this course to run the simulation, and the simulation itself would require time, resources, and a knowledge of Unix-based systems that I do not have right now. Unfortunately, I was unable to find the documentation necessary to create different simulations involving any ligand present at different concentrations, so I decided to run an NAMD simulation of the protein in the solvent box that I created. As NAMD was a topic that was covered earlier in the class, Running a simulation of NAMD was a fairly straightforward process. I created an interactive session with NoMachine and I ran the simulation, but I was still getting errors regarding allocation of computer cores within the cluster. I let the simulation run for a while, and NoMachine eventually got disconnected, so I ended the simulation at that point. Although the trajectory I generated was short, it was enough to generate a small amount of data on the solvated amyloid beta peptide. In my analysis of the amyloid beta peptide, I found that the bond energy stabilized around 50 kilocalories per mole, while the angle and dihedral energy stabilized around 100 and 125 kilocalories per mole respectively. The total energy of the molecule stabilized around negative 70,000 kilocalories per mole as well, and I created some plots using XM Grace to tabulate these results. If I had more time and knowledge, I would properly parameterize and optimize several solutions of caffeine and amyloid beta peptides in a biological environment, run intensive NAMD simulations to observe the peptide's interactions with caffeine, and interpret those results by applying a statistical analysis to see if caffeine really does inhibit amyloid beta oligomer production. I really do think that caffeine could be used in the future as a tool to delay the onset of Alzheimer's disease through this process or one that is similar to it. This project was not in vain though. I learned how to operate at a beginner's level, a Unix-based operating system, and I also got to experience working on Arizona State's computer cluster, which was very exciting. I was also able to leverage several computer programs to apply the concepts I learned in chemistry to large-scale molecules and simulations. Overall, computational chemistry proved to be a very exciting endeavor, and I hope I have the opportunity to become more familiar with the programs and parameterization of such programs in the future.